Good morning once again. Now we will continue our discussion on fermentation media. In the last video, we have discussed a few components of fermentation media like carbon source, nitrogen source, water, vitamins and growth factors, inorganic nutrients, etc. The remaining components of fermentation media we will discuss in this video. Buffers. You are familiar with buffers. Buffers are the components that resist the change in pH. pH affects the growth of microorganisms. All of you know this. When we want high yield of the product, we should maintain all the conditions suitable for the growth of microorganism. Therefore, maintaining suitable pH is very important during the fermentation. The pH that is suitable for maximum product yield, product stability and good growth of the organism should be maintained during fermentation. The pH that allows maximum product yield, that keeps the product stable and that allows good growth of the organism, these three things should be considered. pH should allow maximum product yield, product stability as well as good growth. That particular pH should be maintained during fermentation. When the microorganisms are growing in the fermentation media, fluctuations in pH will occur. Fluctuations in pH means changes in pH. This pH fluctuation affects the growth and product formation by organisms. So, fluctuations in pH should not be allowed. For this, buffers are added into the medium. Buffers are used to control drastic changes of pH. Buffers neutralize the excess of acid or alkali and maintain the pH constant. Suitable buffer is selected based on studies with individual fermentation. Buffers are usually the mixtures of weak acids and weak bases. Commonly used buffers are sodium or potassium salts of weak acids and weak bases like Na2HPO4 and NaH2PO4, K2HPO4 and KH2PO4. Monobasic and dibasic salts. These are used. Calcium carbonate can also be used as buffer. At lower pH, proteins or amino acids can act as buffers. Here we have given the examples of buffers that can be added to the fermentation medium. You can use monobasic or dibasic salts such as Na2HPO4 and NH2PO4, K2HPO4 and KH2PO4 as buffers. In some fermentations, calcium carbonate is added as buffer. While in some fermentation media, we can provide proteins or amino acids as buffers. They can act as buffers. The next component, antifoams. During fermentation, foam is generated. This foam is generated whenever the media has proteins or peptides. If the medium is containing proteins or peptides and we are providing aeration as well as vigorous agitation, it will cause foaming. Foaming is not desired during the fermentation because foaming causes various problems. For example, foaming may lead to contamination. Foaming may create obstruction to aeration. Therefore, it is necessary to remove or neutralize this foam with the help of anti-foaming agents. So, contamination, obstruction to aeration, and there is one more problem with foaming, that is loss of medium and loss of cells. Foaming may lead to three problems: contamination, obstruction to aeration and loss of the medium. And we don't want this during fermentation, and therefore it is necessary to neutralize this foam with the help of anti-foaming agents. There are two types of antifoams: crude antifoams and inert antifoams. Crude antifoams are commonly used antifoams during fermentation because crude antifoams are cheaper. Lower cost is there. For example, corn oil, lard oil, cottonseed oil, soya bean oil, veggie fat oil, octadecanol, etc. All these are crude antifoams. Different types of oils are there: soybean oil, corn oil, lard oil, cottonseed oil, veggie fat oil, and some long chain alcohols like octadecanol. These can be used as crude antifoams. But these crude antifoams are difficult to sterilize because of their oily nature. Steam does not penetrate, heat does not penetrate because of their oily nature, and therefore they are difficult to sterilize. They are added during fermentation when the foaming occurs in a volume just enough to break the foam. Means fermentation is constantly watched, and when the foam rises, some amount of foam is allowed to accumulate, and if a certain level of foam is accumulated, then antifoam is added. Antifoam will break the foam. This is how crude antifoam agents are used. This is done so because excess concentration of crude antifoam agent may be toxic. The second type of antifoams are inert antifoams. Inert antifoams are non-toxic. These are not used as nutrients by organisms, and these are highly effective to control the foam. These are easy to sterilize. Means there are various advantages of inert antifoams. Crude antifoams are difficult to sterilize. These are easy to sterilize. Excess concentration of crude antifoam may be toxic. Inert antifoams are not toxic. Crude antifoams may be consumed as nutrients by the organisms. These are not used as nutrients by the organisms. Although there are many advantages of crude antifoams, they are not used very commonly. They are used rarely, and they are used especially during research. This is because there is one major disadvantage of inert antifoams, and that is their cost. They are costly. The example of inert antifoam is silicon compounds. Antifoams may be added manually or automatically. What is manual addition? Just now I told you, fermentation is watched constantly, and when the foam arises, when suppose 1 feet foam is built up in the headspace, antifoam is added, the foam is allowed to collapse, and the fermentation is continued. It is continuous, it is done so. That is called as manual addition. Automatic addition means what? In the headspace, 2 electrodes are set up, and these electrodes are connected to a pump, and that is connected to antifoam reservoir. When the foam rises in the headspace, contact is established between the 2 electrodes. That gives signal to the pump. The pump opens and the antifoam is added. When the foam is collapsed, the contact between the 2 electrodes is diminished and addition of antifoam is stopped. This is how automatically the antifoams may be added. The next important component of the fermentation is precursors. What are these precursors? Precursors are the chemicals which are directly incorporated into the product, accelerating the rate of product formation and increasing the yield. Precursors, what we provide in the form of precursors is some ready-made functional group is provided to the product that goes and gets incorporated itself into the product, into the structure of the product. And because of this, the rate of product formation is accelerated, increased, and the final yield also increases. Precursors also decide the nature of major fermentation product in penicillin fermentation. Precursors are directly incorporated into the side chain of penicillin structure. Precursors were discovered during penicillin fermentation. It was found that whenever corn steep liquor was added into the medium that is used for penicillin fermentation, there was an increase in yield. This increase in yield was because of phenyl acetic acid supplied through corn steep liquor, and later on phenyl acetic acid was used as a precursor for penicillin fermentation. As I told you that if you use different type of precursor for penicillin fermentation, you get a different type of penicillin. For example, phenyl acetic acid gives penicillin G or benzene penicillin. Phenoxyacetic acid gives penicillin V. Hydroxyphenyl acetic acid gives penicillin X. This is for examples purpose. For vitamin B12 fermentation, we use cobalt chloride or cobalt nitrate as a precursor. For L-lysine fermentation, L-glycine can be used as a precursor. Now we will discuss the last component of fermentation medium, that is chelating agents. Certain medium components interact with each other during medium preparation or sterilization and form precipitate, which makes the nutrients unutilizable. For example, iron, manganese, calcium, zinc, etc. When we prepare the fermentation medium, we take water and we add different ingredients into that water and dissolve it. But when we do so during preparation of medium, two or more components may react with each other to form a precipitate, or they will not form precipitate at low temperature. But when we sterilize the medium by using steam under pressure. So there is elevated temperature, and at high temperature these components react with each other and they form precipitate. If the precipitate is formed, the nutrients are made unutilizable, and therefore formation of such precipitate is undesired. And such components include iron, manganese, calcium, zinc, etc. Chelating agents form complexes with insoluble metal phosphates and other metal ions, and the metal ions are then gradually utilized by microorganisms during fermentation process. Now how chelating agents act then? They form complexes with these precipitated insoluble metal phosphates and other metal ions, and these metal ions are gradually utilized by the microorganisms during fermentation process. Chelating agents preferentially form complexes with metal ions, but the concentration of chelating agents is critical as they may be inhibitory to microorganisms. Means you should not add excess of chelating agents. They may then become inhibitory to microorganisms. The examples of chelating agents are EDTA, that is ethylene diamine tetraacetic acid, citric acid, polyphosphates, etc. Yeast extract and peptones are commonly used as medium components, and these have the ability to form complex with metal ions, gradually releasing them during growth. Hence, there is no need to add chelating agents. If the medium contains yeast extract or if the medium contains peptones, then these themselves are the chelating agents, and therefore there is no need to add any other chelating agent into the fermentation medium. I hope you understood all the components of the fermentation media. Go through this video again and again, and you can ask me if you have any doubts. Thank you.